Hello students, welcome back to your online class. So in our previous video we were discussing plant reproduction. In that we have already discussed about asexual reproduction, different methods of asexual reproduction. We have already discussed about vegetative propagation. So unlike those method of propagation, today we will be discussing another method of propagation which is carried out within a laboratory condition, and that is referred to as tissue culture method. Now name itself suggests to you that here we have a role of tissue to play. So here the new plants is arisen by taking out the small portion of a tissue from a plant. What kind of tissue do we usually take? We take meristematic tissue, because it's only meristematic tissue which brings about the growth in plants. So we will be taking the tissue from either tip of root or shoot, because these are the parts where you have a meristematic tissue. So we take a small portion of a tissue from these parts of a plant and then grow them under a culture medium, artificial culture medium. Now what does this culture medium consist? It's like a food for this tissue to grow. Now this culture medium, it consists of different kind of nutrients and hormones which are required by the plants in order to grow. Okay, so initially this tissue is first put within a laboratory condition, in a laboratory and a sterile condition where the air is pure enough, where no hindrance of any microorganisms in order to rot the tissue is present. Okay, so first you take up the meristematic tissue from the plant and then put it in a culture medium which will provide the tissue with enough nutrients for its division, for its cells to divide. So first the cells of a tissue divides and forms a mass of cell that is referred to as callus. Okay, now this callus further divides and forms a small plant like structure that is referred to as plantlets, and this plantlets further can be rooted in a soil and then it will give rise to a new plant. So here you see that using a tissue you are giving rise to a new plant. This one is very efficient method because since it is carried out within a laboratory space, so you do not need to worry about the space. Even in a small space you get a lot of plantlets which you can later on grow in a soil. Regardless of any kind of season you can grow this tissue because you are artificially providing the tissue with the nutrients. So you do not need to worry about the season that you are growing that particular plant in. Okay, so this is one of the method of propagation, artificial method of propagation which is carried out in a laboratory condition where you have a role of a tissue. We take just the tissue from the plant, the meristematic tissue which helps in growth of a plant. We take the tissue and then grow it in a cultural medium, and then later on transfer the small plantlets that have been arisen from the tissue to the soil, and that plantlets will later on give rise to a new plant. Okay, next we will be discussing about, we are done with asexual reproduction. Now we will be discussing about sexual reproduction in plants. Okay.